Well, so then I go and take over. So hi, it's Gabriel. So right next to me is Xavier from the Schweda Lab in Basel. And today we're going to talk about scoring. Just a second. So it's all about establishing a similarity metric between a model, which in our case is a theoretical prediction of 3D coordinates, and the ground truth for a reference structure, which is typically based in experiment. Now, the goal of this session really is to introduce you to scores that are used by the structure prediction community to assess protein-protein complexes and protein-ligand complexes, and also give you a way of how to compute these scores. So this is not about differentiable error functions that you can directly integrate in your deep learning models. Now, when talking of complexes, this slide already simplifies things because what you see here are only monomers. Now, just a second. So one problem you're having, as soon as you have more than one chain, you have this chain mapping problem. So most scores require to have a one-to-one relationship between chains in your model and chains in your target. In case of dimers, that's trivial. I mean, you can assign them either in two ways, either one way or the other. But this can become difficult if number of chains increases, or if there is different stoichiometries, or if there are even small molecules involved. Now, the good news is OpenStructure, the tool that you will use later on to do actual scoring, can deal with this in the background. But you still need to be aware of this issue that we need these assignments for scoring. Now we can directly jump into actual scores that are popular in the field. So I would like to start with LDDT, the local distance difference test. And it is actually based on extracting a set of reference distances. So basically all pairwise distances in a reference structure that are within specified distance threshold. And then LDD just reports the fraction of these distances that are similar in the model. So LDT was originally designed back in the days for comparing tertiary structure models, so monomers, but it can easily be extended also to oligomers. So that's why it can be used for complexes. And one important feature you see here is that the two models, also the ground truth and the model, are actually quite dissimilar. However, LDT identifies kind of conserved substructures that it predicts to be similar. And in fact, it's two kinases, one in a closed and one in an open conformation. And LDT only penalizes the distances in between these rigid substructures and still gives the structure a relatively high score. Various scores like an RMSD or God knows what would already have trouble to identify that these models are really, really similar. Now, here is a much more extreme case. In white, you have a reference structure and then a model colored by local LDT values. And LDT thinks this is actually a pretty good model, even though there's no way this superposes nicely globally. So you would not immediately see that by something like RMSD or other scores or GDT or TM score that are used. But if you just split the model into two, so basically you cut this flexible linker region here in the center and superpose the domains individually, you see that actually the model is quite well representing the target. It's just about this flexible linker region. Now, the split of structures has been done in previous CASP experiments manually. To split structures in so-called evaluation units to mitigate this effect of domain movements, to then score them with these superposition-dependent scores, primarily GDT or TM score. But in a fully automated evaluation pipeline, you don't want to do this you don't want to do this manual splitting. So that's why we prefer superposition independent scores like LDT or CUT that are less affected by these issues. Now, the next score I'd like to introduce is QS score. It is similar to LDDT in a sense that it also operates on pairwise distances, but has two key differences. One of them is it only operates on pairwise distances across the interface, so it's very specific to evaluate protein-protein complexes. And the second difference is symmetry. Now imagine you have a model that has a completely wrong interface. Now, the problem with LDDT, the distances you're evaluating are solely extracted from the reference structure. So if there are so-called added model contacts in the model, they will never be checked by LDDT. So added model contacts actually do not penalize LDDT score, whereas in QS score, all pairwise distances within 12 angstrom in any of the two structures, so model or ground truth, are considered, meaning it does not really matter which one is the reference and which one is the model, so it's symmetric. That's the second key difference between LDDT and QS score. Now, the last score regarding protein-protein complexes is DocQ, which is strictly for dimers, meaning protein complexes consisting of two chains. And it's actually based on three metrics that are used by the Capri community for decades now. So that is FNUT, L-RMSD, and I-RMSD. And then this Capri community defines that a classification of models into incorrect, acceptable, medium, or high based on the outcome of these three metrics. Now, FNUT in principle is really just the fraction of reference contacts that are recovered in the model. Whereas then we have two RMSD measures. So there is this ligand RMSD, L-RMSD, where we define dimer as one chain is the receptor chain. It's just by definition the larger one. And then we have the ligand chain, which is the smaller chain. So we perform a superposition on the receptor and then just see whether the ligand ends up at a similar location, and we measure the error as an RMSD. Now, the last thing, the interface RMSD is a bit more localized. So we identify the interface by just saying, okay, each and every residue that is within a certain distance of the interface is considered also within a certain distance of another chain is considered an interface residue, and then we compute an RMSD solely based on these residues. So it's a bit a more localized RMSD measure. Now, DocQ then is and then one important point we want to mention here is again this local superposition issue. That mainly affects this ligand RMSD, because just think of these huge disordered regions you sometimes see in AlphaFold models. Now imagine you have such effects in your receptor, so this really kind of distorts your superposition because it's a minimal RMSD superposition. So you can have large effects of either just domain movements that result in a poor superposition, or you can affect of these disordered regions that just make your superposition go nuts because it's so susceptible to outliers. Now, DocQ then is nothing else than a way to combine these three metrics into one continuous score. And this continuous metric then really happens to nicely separate these four classes of the Capri community. And it just behaves a bit nicer for the machine learning crowds to have this one value they can train for. And with this, we can move on
to the ligand scores, which I will be talking about now. So basically, imagine that we have a protein and there's a ligand, a small molecule that binds to this protein. We can define a binding site with some definition. The exact definition doesn't matter, which is the region of the protein which is close to the ligand. And then we're also interested in seeing how are the interactions between the ligand and these binding sites conserved in the model versus the reference structure. And so in OpenStructure, we have 3 scores to look exactly at these 3 things. So the first is the busy RMSD. It's symmetry corrected ligand RMSD after binding site superposition. So you superpose the binding site and then you look at the RMSD of the ligand. This gives you a score which is between 0 and infinity. Infinity is not very nice, so usually people simplify it to binary success or failure and define a success as a prediction with an RMSD typically smaller than 2 angstroms. So this score depends on a superposition. It's a local superposition, but before looking at the score and trusting the score, you should look at the RMSD of the backbone superposition and make sure that the superposition was successful. And yeah, so this score is very sensitive to outliers because it's an RMSD, so it goes in squares for some of the and this can be a problem with weakly interacting large ligands, especially with a loose part of the ligand that can have a very large effect. The second score that we have is LDDT-PLI, which is the LDDT of the protein-ligand interactions. It's a score between 0 and 1, which represents the fraction of the conserved protein-ligand contact distances. So similar to LDDT, we establish distances in the reference structure, and we look how close they are in the model. And that's really only the contacts between the ligand and the binding sites. This score is superposition independent. One issue that it has is that it goes towards 0. It tends to 0 pretty quickly if the ligand is posed outside of the binding site. Yeah, so that is something to keep in mind if the score is 0. And finally, we can look at the binding pocket or with the LDDT-LP or the LDDT ligand pocket. Again, it's a score between 0 and 1, which represents the fraction of conserved contact distances within the binding site only, not looking at the ligand at all. Again, this is a superposition independent score, which looks at the binding pockets. And so basically with these 3 scores, you're really able to look at all the aspects of your prediction, both the ligand, the binding pocket, and the interactions between the ligands in the binding pocket. And all these scores are provided by OpenStructure. There's one thing that I mentioned is that the scores, and especially the RMSD, but also the LDDT-PLI are symmetry corrected. What does that mean? When you have a ligand molecule, you often have atoms which are chemically equivalent. So if you think of the 3 oxygen atoms that are linked to this phosphate, this is simply an ATP molecule. These 3 oxygen atoms are actually chemically identical, and they could be any of these 3 atoms here. So when you compute a score, you need to take into account that you don't have a fully clear one-to-one mapping of the atoms. And so what we do when we do a symmetry correction is that we actually match these 2 molecules with graph isomorphisms. And this gives us basically mappings between atoms. And to establish these graphs, we consider the element of the atoms and ignore bond orders. So this is something to keep in mind. If you were to predict the wrong elements in your ligand, we would not be able to find isomorphisms between the molecule. So it's really important to predict the correct atoms. And again, we ignore the bond orders. And so we've been talking about open structure. So now a few more words about it. So open structure is an open source computational structural biology framework that we developed at the University of Basel and SIB, Swiss Institute of Bioinformatics. It contains a lot of I/O functions and algorithms to handle and analyze structures and sequences. And in particular, what's of interest here is that it contains a comprehensive framework for reference model scoring. So all these scores that we've just shown, they are available in OpenStructure. And so it's available in two flavors. It's a Python package. And here I just show some example code of how you can use OpenStructure. Basically, you start with import OST to import OpenStructure, and then you can play loading data and running the algorithms. It's also available as command line with a command line interface, and we call them actions. And in particular, we'll see in a moment the OST compare structures action, which allows comparing and doing reference model scoring on the command line. How to find help with OpenStructure? So we've added a lot of links of where you can find out more about it. There's our homepage. The documentation is available here. You can also get the documentation directly within Python with the help command. So this works well. And so the source code is available on our GitLab here at the University of Basel. And we also have mailing lists to basically a help desk mailing list to help out users and developers. And so how do you install OpenStructures? There's three main ways you can do that. The first is to install from source on your Linux machine. We have a full documentation on the homepage. It's quite difficult. There's lots of dependencies, so we don't necessarily recommend that for everyone. Instead, we provide containers with Singularity and Docker, which are much easier to use. You just have to install the containers, and you can get things running very quickly. So this presentation is going to is already available on the GitHub website repository of this workshop, and so you'll be able from the PDFs there to directly click on the links and get to the relevant pages. So don't worry about it. And the last way that you can use OpenStructure is through Conda, and this is what we are going to demonstrate right now with a hands-on session. So I will just quickly interrupt the presentation here to make sure that everyone was able to get to the website of the repository and download and open the Colab workflow so click on this link here for this session, metrics for measuring proteins and complex model accuracy. And once you've done that, this will open a Colab notebook on Google Colab. I just need to reconnect here. Please make sure that you've run the first two the first two cells while we're talking about going through the example. I will go back to the presentation, but please make sure that you run this, and because they take a few minutes to run, and it would be good if we can continue right away. So let me continue with the presentation. There's a question? Right, and if you have any question, don't hesitate to use the Discord and ask your questions on Discord. If you get into if you have any trouble running this Colab notebook. And so in the hands-on that we're going through in a minute, we have two examples. One which is more Pinder related, which is a dimer scoring example, and one which is more Plinder with a ligand scoring example. So the Pinder example is a Cas15 target. It's tobacco lectin, which is also available on the PDB. And so we are going to compare this reference structure to a Cas15 prediction from the participant Delft lab. And the second example that we're going to run through for the ligand scoring is a Cameo target, which is also available on the PDB. It's the CHEK1 kinase with imidazole LRKK2 inhibitor. And here we are going to compare this reference structure with a model that was obtained with Swiss model, and then the ligand docked with Autodoc Vina. And with that, I will get back to the notebook. What happened? This is not what I wanted to do, I'm sorry. Yes, Gabriel is going to run you through if there's no, is there any questions on?
Yeah, so Jay is very busy in answering the questions, so it's quite a hard time of figuring out which question is not yet answered.
Yeah, no, but I think it might be worth having a bit of a break before the notebook, because there's quite a lot of questions.
Sure.
Okay, so let us take the questions.
So I think one that's actually quite interesting, which maybe we can go a bit in detail, is for LDDT and LDDT-PLI, what is the meaning of the metric? Does it have a real meaning? And how do you correlate the number to the accuracy of the prediction?
So in the end, there is no real, let's say, biological or chemical meaning. I mean, it's a geometrical comparison in the end, so it's really a similarity metric.
Yeah, no, it's a similarity metric. One way you can interpret it, if it's 0, basically it means that for LDDT-PLI, right? So if it's 0, it means that you post the ligand outside of the ligand pocket. As soon as you have something which is above 0, it means that there's at least some parts of your ligand which are within 4 angstroms or within 4 angstroms of the gold standard. So then what is, I mean, and then basically it increases the more accurate it is. But yeah, you're right. There's no real
biological meaning. So Xavier mentioned this, or threw in the room the 4 angstrom value. So 4 angstrom is really a distance difference threshold. So you have your reference distances, you have your model distances, and you basically just count the number of distances that are within a specified threshold, which, for example, is 4 angstrom. Now LDDT, the whole LDDT score does that for a very accurate, so a very small distance difference. That's 0.5 angstrom for 1, for 2, and for 4 angstrom, to kind of cover a bit this whole accuracy spectrum. Now, what's another effect of this is you kind of avoid a little bit this outlier behavior you would have from RMSD. So at some point, you're wrong. It doesn't matter how wrong you are, you're just wrong. So that way you kind of don't have this huge effect from outliers. So that's a bit more details in the behavior of LDDT. But yeah, I'm just shooting if there is anything more.
So there's a few more questions I see in the chat. Is ligand still needs to be done against reference? how do you select a reference? So yeah, in all the examples that we are looking at here and for Plinder, we always compare against a reference from the PDB, which is the protein target with the ligand target, which was given in the target. So we always have a reference, and it's, I don't know if it's clear from the chat, but it's not whether it's a apo or holo, it's really you're comparing a prediction with a ground truth, and the ground truth is containing the one containing the ligands.
There was also just this question again about the LDT, LDT-PLI, just to finish that answer on like how to understand the number. So what would you say is a good prediction for LDT, for also interface LDT and so on?
Yeah, so that is a long discussion. So what has settled a bit for what we are using, at least in Cameo, when we do evaluations of how good are people in predicting LDT, we do ROC analysis, so we have the exact same problem, but it's good, but it's bad. And so in the field, it seems like 0.6 is kind of this sweet spot of where we say, hey, this starts to be a good prediction. Because what you have to consider is LDT. I'm not talking of LDT-PLI now, just general LDT never really goes down to 0. Because from neighboring residues, you always have a few distances that are conserved just by pure chance, because the residues are close and you don't have a possibility to have too large distance differences. So that means in the range 0.2, 0.3, you're in the random range. But as soon as you come up to 0.56, then you start to be pretty good. Then on the other hand, LDT-PLI, as Xavier said before, so as soon as you're a bit away from the binding site, you drop to 0 quite quickly. So if you have a non-zero value, this means you are in the correct binding site. So just a few notes on that.
Right. And so I see there's one question from Rashid. Is the PLI LDT calculated against all the pairwise atoms in the protein residues with the atoms in the ligand, or is it computed for one specific atom per protein residue, for example, the alpha carbon? No, this is a all atom in the both in the protein and the ligand. So really all interactions within 6 angstroms of the ligand in the protein, so all atoms.
But important to note here, only interactions between the ligand and the protein, not within the protein.
Right. And yeah, so that is
Xavier, if you, yeah, exactly. Okay, you got the question. So just about the 2 angstrom.
Be good. In the end, it's all about being better than the others, right? So if you can, if your RMSD can be better than other methods can predict, then I would say it's good. And if not, then it's not good. I don't think there's a magical value at which a prediction becomes good or bad. It depends on the difficulty, and you've mentioned, Jay, the difficulty a bit earlier. Yeah, I would say different difficulties are going to warrant different analysis.
And just to also reiterate, right, like the RMSD, that's of the ligand, right? So that's not about the interactions. And that also tends to be skewed a bit by, for instance, the size of the ligand. If there's like a part of the ligand that's floating around that's not interacting with the protein, how important is it to get that part right? So that's kind of why you kind of need not just an RMSD when you're trying to predict both the protein and the ligand.
Good. I think we've addressed all the questions so far in the chat. So maybe we can start with the Colab notebook. I hope everyone was able to get it up and running, and Gabriel is going to take over after the Mamba install that we've done.
Good, right. No, so this is really just to check whether it's installed correctly. So
wait, are we still sharing the? Did we stop sharing the screen?
Jay,
Yes.
do you see anything? Good.
No, you have not shared the screen.
I'm sorry. I think I clicked on share. Right. I'm sorry about that.
Much better. No, so this command here is really just this ost ---- version is just for those who are not familiar with Google Colab, with exclamation mark, you kind of do a command line argument. You just check, is open structure around? And yes, it is. And you can do the same just by importing the Python module. So we're operating with open structure 280. So not too much on the details here. So we have a data loading section. So this is just prepared by us. So we have some structures to play around, and we wget some of the structures. They will end up in your current working directory on the left, right? Right, so they're here in this directory. It says T1187. So you can open it and you see basically two structures that are present. Now T1187, for those who know COSP, immediately recognize this as a COSP target. So this is the example that Xavier introduced. It's a dimer, and we will see how good this model actually is. First, by using OpenStructure interactively, so use it from Python, so you can then basically copy-paste this in your for loop or in your evaluation pipeline. So we're loading structures just with this IO module that also has been very briefly introduced. So we have now the OpenStructure representation of these structures. And then there is this scorer, whatever that means. So the score itself is kind of a wrapper around many scores that we just presented, but also other scores that are relevant for the field. So people have heard of GDT, people have heard of TM score, RMSD, etc. It's all in there, it's all available. And what we're doing here is really just run this code block that should just exemplify a bit what we can do with the scorer object. So once you have constructed it, you can just do a raw help, and you will get a huge docstring that tells you what is available. And just from an implementation point of view, all these scores are available as attributes that are lazily evaluated. So meaning once you computed them, they're just stored in the scorer object, and you can access them again in their cached. So that's just from an implementation point of view. And of course, I'm not going into all the attributes that are available, and I guess that's something for you to explore. What can we do actually with this? Because this will be way out of scope. Now, the example here is Simple Compute's LDDT and QS core. So we see the model is actually very accurate. I told you 0.6 LDDT is already pretty good. We have the backbone-only version that operates in C alpha atoms. I suggest you use the all-atom version, as this is what LDDT is supposed to do. And then there is this QS core, and again, everything above 0.5, 0.6 is really, really good. And if we use this visualization, we can also basically see, so this is really just two structures that we just loaded. Well, I have a bit issues with this Mac. I'm not so, yeah. So what you're seeing here is literally, it's two chains are nicely superposed. Mm-hmm. That's actually a perfect model. There is the chain mapping that I mentioned. So I told you that OpenStructure does this fully automatically in the background. It does so by enumerating all possible mappings of the model onto the reference and optimizes for best possible QS score. You can do that for, let's say, up to 8, 9, maybe 10 chains, but then it starts to be really computationally expensive because the number of possibilities to map these model chains explodes with factorial complexity. And as soon as you go larger, OpenStructure implements a heuristic that is not guaranteed to give you an optimal mapping, but typically is pretty good. And then there is also DocQ. Now, DocQ is strictly timers. So this is why you basically get a list of pairwise interfaces, and all the DocQ values are then here also as lists. So you evaluate each interface individually. There are also ways, if you check in the documentation, to kind of combine these into average scores, etc. Now, as a second way of running these things is this compare structure action that Xavier mentioned. So this is really just a kind of a command line tool. And if you run this, you see this kind of minus out. You have to refresh the file browser, and then you see this JSON output that popped up, and you can see what kind of information OpenStructure spits out. So again, you have your chain mapping, you have the alignment between the chains, you have all the scores, the ones that you already computed before, etc., etc. And there is more to be explored. So there is, you can also just run it with OST compare structures -h to see what it actually can do for you. And with this, we can move on to the ligand example.
So I was asked before that to check if there's people in the waiting room for Zoom. How do I do that? It's here. I don't know how to do that. I will assume that there's no one in the waiting room. Okay, tell me. We have no waiting room. Yes. Okay. Very good. Thank you. Right. So let's continue straight. With the protein ligand complex scoring. And so you will see we are going through fairly similar steps. First, we have to download some data, which we made available in the repository. And here I will also. Oh, I don't even need to refresh. The data appears here in a separate folder. And again, so this is quite similar from what you've seen before. We will now load the data into OpenStructure. Here, the only difference that we have is that it's in addition to a chain in PDB format, you need to load a ligand, which is typically done from, and that's also the case in Plinder, which is loaded from an SDF formatted file, and this is with the load SDF function. And yes, so this I run. Now, a slight difference from the protein scorer that we've seen before. For ligand scoring, you do need to do a little bit of cleanup before running the scoring in OpenStructure. And really, all you need to do is to remove hydrogens. Because if you have hydrogens, then we're not going to be able to properly match ligands with isomorphisms. And so all this does here is to remove hydrogens first from the model and the reference chain structures, so protein structures. And for that, we use a function called MOLC, the molecular checker from OpenStructure. And all this does is to remove unknown atoms and hydrogen atoms. And so then you have these clean structures. And for the ligands, we can simply do this with a selection where we select everything which is not hydrogen or deuterium. And why deuterium? Sometimes in the PDB, the structures come with heavy hydrogens, which are deuterium atoms instead of hydrogens. And so by saying La not equals H and La not equals D, you make sure that you've eliminated both cases. Typically from the models that you predicted, you would only need to remove hydrogens. It's unlikely you would predict the deuterium atoms. And then we can run the ligand scoring interactively in Python. And as we've seen before, so you can follow this link here for a full documentation of all the functions and features of this and options of this scorer. So we have two scorers here, one to compute LDDT PLI and one to compute the busy RMSD. And they work very much similar to the protein scorer. You give them a target structure, a model. In addition, you need to give them target and model ligands. So that's the ligands from your target and from your model. And so if we run this, basically what happens is that we will get a scorer object with a score property. Because there can be more than one ligand in a model or in a reference, you need to address these ligands by chain name and residue number. And basically these two lines are simply getting the chain names and residue number that you need to properly index the dictionaries in Python. So this gives you the LDDT PLI, and for the busy RMSD and LDDT LP, it's similar with the ST-RMSD scorer, and busy RMSD is in this .score property. LDDT LP is an extra information which is available a bit differently. You need to get this aux or auxiliary dictionary objects. You index it the same way as the score, but then you get a dictionary with a lot of information, and one of them is the LDDT LP. I should have put the visualization first. So let's go to the visualization. So here we do something very similar to before. We use Py3DMOL. And so there's one more step which is here, which is basically the superposition of the model onto the reference structure. And this we can do with OpenStructure. So there's a syntax that you just need to copy paste to do the superposition with an editor and then apply transform. And the superposition is available also in this aux property under the transform, and this basically applies your superposition, and then we save this data for the viewer and load them in the viewer. And you can see that two files have appeared here, and these are the two superposed files that we're going to see. And yeah, basically, so here's the result, and you can also see. So we have in this case a very good prediction, not only for the protein. You see that the binding site superposes almost perfectly, but also the ligand, which in green is the reference ligand, and in gray and colors is the model. They are also very close to the gold standard reference structure, and this is reflected basically in the scores. And with, you see there's the busy RMSD is below 2 angstroms. This is a success. It's not, you can see that it's not absolutely perfect, but it's very close. And similarly, basically the LDDT PLI is almost, yeah, above 0.75. So this really means that most contacts between the protein and the ligand will be satisfied and conserved with the correct distances. So that's again a very good prediction. And finally, we can run exactly the same things through the command line. Here we just need to use a different action. It's to compare ligand structures action. It's very similar to the compare structures action. There's just a few more arguments where you need to give the reference ligand, the model ligand, and basically tell it which scores you want, LDDT, PLI, and RMSD. And now that I run this, I need to refresh this to see the output, which is appearing here. So yeah, basically you also get a JSON output from this action, where you will see a list of the ligands that you gave, and then there's LDDT PLI with scores and lots of extra information, and then BZ-RMSD, which is abbreviated as RMSD, with again a lot of information. The score, you see, this is the same score. So this is literally the same information that you would get from the scorer object, but you can run it on the command line. And yeah, that's it. That's all we had in store for you. Are there any questions in Discord? I think there are plenty. I see lots of activity. MMSIF structures or PDB, yes. So you can absolutely load MMSIF structures. If I go back to the presentation, I think you have an example here where you can, so this is how you load the MMSIF file, is with load MMSIF instead of load PDB or load SDF. And then for the ligands in the PDB, you can then have them as MMSIF, and you don't need to give them as extra arguments if you give an MMSIF file. Just be aware that for many of the use cases that we have, actually for most use cases in Plinder, the ligands will be known, and so they will already be in the PDB, and so that will be fine actually. If you venture a bit outside and you have ligands which are not in the PDB, then MMSIF is not necessarily the best file format to represent those ligands, because it uses a compound dictionary which has only a fixed set of ligands. And there's another question. If the prediction has a slight different residue dictionary, like protonated proteins or histidine tautomer, is that considered already in Plinder for alignment? Yes, so LDDT also considers the tautomers, and it's also a sort of symmetry correction. On the protein side, it's an easier sort of symmetry correction because it's just two atoms to swap. But this is considered also on the protein side. And protonated proteins, yes, so the scorer object for protein-protein interactions removes hydrogens. So this cleanup is part of the yeah, this is part of the scoring. It's just for the protein ligand scoring part, the protonation. Hydrogens are not removed automatically. Missing in the example case for LDDT PLI, we can see some residue missing in the loop near the binding pocket. How are these scored? Let's get back to the Colab. So you see, well, I'm not sure exactly what the question is. So so in this, oh, this loop is missing. Yes, so LDDT doesn't penalize, and so okay, so I forgot to mention here. So here in yellow is the gold standard, and in purple is the model. LDDT doesn't penalize missing residues in the reference structure. So if they are missing in your model, they will be penalized. But if they are missing in the reference structure, then this is not penalized. And so yeah, you would still be able to obtain a perfect score if your prediction was correct. So while we're waiting for more questions, there's a few more things that we wanted to say before we close the session. So in summary, we showed how to compute LDDT, QS score, and DocQ for protein and protein assembly scoring. We highly recommend that you prefer superposition-free scores in automated assessments, such as LDDT, QS score. There's also the CAD score, to mention just a few, to avoid issues with superpositions of more than one domain and flexibility in the protein. For ligand scoring, we showed you how to compute LDDT PLI, BZR-MSD, and LDDT LP. And all these metrics are basically the metrics typically computed in benchmarking experiments such as CASP, Cameo, and we showed you how to compute these with a notebook. So we highly recommend you go through this notebook and you can really use it as a starting point for your work. We wanted to cover a few more things, right? Typical,
or just keep it as future reference. I mean, Jay needs to.
Yeah, do we need to
have a look at the time?
No, there's plenty of time. Please.
There's plenty of time,
Yeah.
so we can quickly go through a few more things that we thought could be interesting. So of course, often enough, especially when you start creating a prediction methods, you will run into issues and your scores are going to be low, and you're going to wonder, why is my score low? And so here are a few things to check, typical reasons that we've seen in the past for why a score would be abnormally low. So the first thing to check is check your residue numbering. It might be somewhat surprising to some, but in the structure prediction field, residues are numbered in a one-based format by convention. So the first residue is 1 and not 0. So just be aware, we've seen very often very low scores because of that.
Just due to misalignment, also important there is if you use the scoring interactively, there are flags where you can say, hey, I want residue number alignment. So basically you define your alignment by residue numbers, or you perform Needlemann-Wunsch. Preferably in benchmarking cases where everything is nicely numbered is residue numbering.
Thanks. Then the next thing to do is check your superposition for superposition dependent scores. There's quite a few that we've seen. There's DocQ and the BZR-MSD. And so, I mean, of course, for global superpositions, that can often be a problem, but also local superpositions, I mean, they sound really simple on the paper, but they can often be wrong, and that can lead to surprisingly low scores. So check your superposition and check how this happens. A few words about processing in OpenStructure. So when you load a structure in OpenStructure, in order to have it loaded properly in a way that's useful, there needs to be some processing which converts just atomic coordinates into really a protein which is loaded in memory. And a few things to keep in mind, that there's a compound library which contain residue and atom names, and this we obtain basically from the PDB, and it defines intra-residue connectivity. So if you don't properly follow the naming of the atoms in your files, you will have problems and low scores. If you don't follow residue names, so for the amino acids on the protein side, you will also have issues or will not be able to connect the protein, and this can result in basically parts of the structure being removed. There's also connectivity which is made for polymers to basically bind inter-residue and peptide bonds, which are they are implicit in all of the PDB, and if your model is a bit not so great, this can sometimes fail and lead to really low scores, even if visually you would think this is not so bad. One other thing to keep in mind is stereochemistry checks. Some flavor of LDDT penalize for serious stereochemical violations. Basically clashes. If you have a clash in your protein structure, the whole residue, both residues which are clashing with atoms that are clashing will be removed, and this can also quickly lead to scores going down very quickly. So this is the case for LDDT.
And it is reported in the output, in the JSON output as well. It's accessible through the score objects when, I mean, it's really serious stereochemical violations, and they are reported.
And yeah, so just for ligand scoring, LDDT PLI and LDDT LP do not perform the stereochemistry checks. So yes. And then for ligand scores, check if the ligand is unassigned, and in the JSON output, there's gonna be some hints of possible reasons why we couldn't score the ligand. Typically, it's because you predicted the wrong ligand, or because it was too far from the binding site. So there's gonna be a few hints of why you don't get a score in the JSON outputs, and also in the interactive scorer object also has this information. Then why is it slow? Do you want to?
Yeah, so one issue you can run into is if you have huge complexes with a lot of chains. I mentioned that this factorial complexity, it's just the number of possibilities to assign model chains and the target chains. There is a heuristic in place that brings you quite far. So for Pinder use case, it's absolutely no problem if you have timers. So that's not an issue. So the heuristic brings you up to 24 MERS or even further to 70 MERS if you're okay with waiting for an hour or two. Yeah, then for the ligands, it's
so there's also a few reasons. So maybe so the first, and it's especially if you have a large number of identical ligands either in your structure and your model, or your model and your model. One thing to keep in mind is we do an all versus all scoring. So all scores are computed before choosing the best one. And yeah, of course, with a large number of ligands, this can be an issue. Very symmetrical ligands, we iterate over all isomorphisms of ligand atom mappings. This is typically not too much of a problem in most common ligands. There's going to be a few parts of the ligand which are symmetrical. There's going to be just a few isomorphisms, but you will suddenly end up with a very highly symmetrical ligand, and you will have then an explosion of the number of mappings which can make everything slow. So if it's a problem for you, you can exclude these ligands which are too symmetrical. And finally, ligands that are in contact with multiple chains are typically a bit more computationally intensive, because we have to run over all the chain mappings to find the best optimal chain mapping. And so if there are more chains in contact with the ligand, of course, there's more chain mapping to compute. So yeah, but in general, I should say typically scoring is really only takes a few seconds, and you've seen in the notebook, it's really fast, and so we hope this should never be too much of a problem. And I see that we have questions on Discord.
Yes.
That's great.
Maybe I can bring up a few
Sure.
ones which are, yeah. So one that's, yeah. So what about covalent ligands? How does LDDT-PLI deal with those?
Right, so LDDT-PLI, the way this is done now completely ignores that ligands are covalent, which could be kind of a problem. So you will, of course, you will see the covalent bound as one contact in LDDT-PLI, but this is going to be one out of dozens of contacts. So it's how should I say? It's handled in a way that you can score covalently bound ligands, but we don't weigh it very highly. I hope that answers the question. Also, is there any limitation with large ligands like sugars or peptides?
You only do non-polymers.
So the scoring is only for non-polymers. In Plinder, there's been quite an effort to convert small peptides and some sugars, I think, to non-polymer ligands or small molecules that can be handled the same way. But if you're using the PDB, you will need to do some pre-processing to score them. If you're using Plinder, then this is going to work out of the box. And of course, if you have larger peptides, you start to have the risk to run into more symmetry corrections. There's more contacts. It's bigger. It might be a bit slower, but there's no strict limitation with that.
So that being said, completely independent from Pinder or Plinder when peptide scoring, I mean, you can just also treat it as a protein complex and use the scores that are available from the scorer in principle. So there is, for example, if people are interested, for example, in peptide modeling, there are also Capri cases where they have some special treatment of peptides, and these special treatments are implemented in the just protein scoring class. So Control F Capri, and you will find it. For saccharides, we didn't put effort in this, so this is, I'm not sure how this behaves.
So say it again. I was looking at the chat. I'm sorry.
Yeah, no, there was also saccharides mentioned, and there is nothing we can do with them.
So yeah, again, in Plinder, they've been converted. At least I think some of them have been converted to be like small molecules. So you would see them as small molecules.
Okay.
Okay. Any more questions?
How do you link prediction with reference? Same name, PDB codes. Does it need a label? So I assume that this is about mapping of ligands between reference and model. You want to answer this?
I'm not sure I understand the question. So can you read it again? Where was it? I don't.
It's a very news one. How do we link prediction with reference? Same name, PDB codes. Does it need a label?
If this is about more from the leaderboard perspective, like of if you make predictions, then evaluating them, basically you have both the reference and the prediction, so you know which ones map.
Oh, I think maybe the question also refers to for ligands. So I mentioned symmetry correction. So this is basically how we map ligands between the reference and the structure. So we transform them to a graph, and we see if the graphs are isomorphic. So in SDF, there's no residue name, so we cannot use residue names for that. I hope that answers the question.